Welcome to a special episode of Performance Around the Clock. In this episode, I will talk to Professor Greg Lemke, who is the Chief Scientific Officer of the Salk Institute. He will talk about his childhood in a small town in Ohio and how he went from there to MIT to do his undergraduate education. And from MIT, he came to Caltech for his PhD, and he did his postdoctoral training at Columbia University before starting his lab at the Salk Institute. Greg Lemke is famous, well recognized for his seminal work in neuroscience, immunology, and also neurodegenerative diseases. All three aspects relate very well with human performance. But recently, he has decided to step down from the position of Chief Scientific Officer and also to close his laboratory. But he's pursuing something very exciting and is in the next chapter of his life. And stay tuned towards the end to hear about his new passion. So welcome, Professor Greg Lemke. Welcome to another episode of Performance Around the Clock. I'm super excited to have my esteemed colleague, Professor Greg Lemke. Professor Lemke is my ideal scientist, and you'll quickly know why, because his science spans multiple aspects of human health, and he is also a well-rounded person with many different interests. So before we begin, again, welcome to the podcast. Welcome, Professor Lemke.
Thank you.
Oh, Greg. So we always start with, scientists have a very different, very interesting journey into science. And I gather that you were born in Ohio, in a small town. And how did you get interested in science? And
I don't know. As a kid, I was always curious about how things work. That's how I got into science. I don't have any. There was no scientific background in my family. My father was a railroad lineman.
wow.
You know what that is?
Yeah.
Somebody who climbs up on poles and fixes telegraph and telephone lines. That's what he did. And my mother, before she was married, she was a hairdresser. So there was really no scientific, as I said, scientific training or interest in my background. I'm actually the first person in my mother's family to go to university. My father's family, I had one uncle who went to university, but other than that, I was the first in his family to go to university.
Wow.
So,
yeah, actually, my grandfather was also in Indian Railway, but he was not climbing the pole. He was keeping track of the goods train coming and going, and mostly in the evening or night shift. So I actually grew up next to my maternal grandfather. So I was fascinated with trains from very early on.
well, yeah, keeping track of things in those times was actually dependent on telegraph communications.
Yeah.
And so, what my father did was actually important to make sure that the trains were running on the right tracks and were running on time.
But then I would have suspected that you would be more interested in engineering, physics, math. And how did you tilt towards biology?
Well, I didn't really know exactly what I wanted to do. I was actually, when I was a kid, as I got into high school, I was interested in all things. So I was interested in physics and math. I did a lot of physics and math. I was particularly interested in chemistry. I was as a kid, I was kind of a pyromaniac. And so I was interested in the chemistry of what made things explode. So that was one of my interests in science. I was interested in explosive reactions and fire.
Well, like kind of see the relation between your interest in fire and then looking at the immune system. It's almost like the cells are on fire when there is immune.
Well, that could be. That's a later story. I mean, we got into the immune system really by accident. We were led into that by the science.
Yeah, so that's why it's really interesting to see how the career of a scientist takes various turns. So you went to MIT, and then was it a little bit difficult to come from a small town in Ohio, getting into MIT, adapting to the new culture of?
Well, yeah, it was a small town. The town was very isolated, so it was 3 hours to any big city.
3 hours by drive.
3 hours driving.
There was no airport, just to make sure.
No, there's no airport. The town had like 7,000 people, and it was actually a pretty good sized town for the area. It was a rural part of west central Ohio. So yeah, it was a big change going to MIT. I mean, I'd never lived in a city. Certainly, I never had exposure to that kind of intensity of education. I mean, I think a lot of MIT students have exactly the same experience. They're the smartest kids in their high school. Everybody tells them they're, great stuff and they're brilliant. And then they come to a place like MIT and everybody's the smartest kid in their high school and brilliant. So it was somewhat of a difficult transition, but I actually, it wasn't that difficult, I would say.
But during that time, so you did your major in biology, or? I
majored both in biology and in chemistry.
Biology and chemistry. Any minor?
Yeah, my minor, well, everybody has to have what's called a humanities concentration at MIT. So my humanities concentration was in music. And MIT has a quite reputable music department. And so my humanities concentration was in music.
Music, performing or playing instrument? What else?
I both did some performing, but I really thought I wanted to write music. So it was actually a very interesting thing how I got into that. I mean, MIT is in Cambridge, which is right next to Boston. It's an incredibly active musical environment. There's many conservatories in Boston that are, top ranked. So there were great music students. I lived actually in a fraternity in Boston, which was physically very close to the New England Conservatory of Music, to a musical school called the Berklee School of Music. It was literally on the same block that I lived. And it was very close to the concert hall, to the Symphony Hall, where the Boston Symphony played. So that was a very important thing. And then the music department at MIT was actually also very good. The first courses that I took at MIT, there was they were run by a professor named Stephen Erdely, who actually became a big influence on me. And Steve Steve was a violinist by training as a performer. His wife was a concert pianist, and Steve had been a violinist in the Cleveland Orchestra with a legendary conductor named George Szell. So he was in the 1950s and 1960s. He was one of the top conductors, and the Cleveland Orchestra is still to this day one of the finest symphony orchestras in the world. So Steve was a violinist in the orchestra, but he was Hungarian by birth, and he was interested in how music is made around the world in different cultures and different environments. And that's part of actually a Hungarian tradition. So there's a famous Hungarian composer named Béla Bartók, and another Hungarian musician named Zoltán Kodály, who had made the study of what became a branch of music study called ethnomusicology. That is to say, the study of how music is made around the world. And so while Steve was a violinist in Cleveland, he got a PhD at Case Western in this discipline, in ethnomusicology. So, and his first job, when he decided he was going to be an academic, was at the University of Toledo, which is not far. It's about 90 miles from where I grew up, north of where I grew up. And so when I took his class, you know, we made this connection, and he sort of identified with me. He was a big, big influence on me in terms of music.
Yeah. Any similar influence from biology or chemistry, MIT?
You know, not nearly as profound. I mean,
did you TA or do something for?
I had to TA. Well, I TA'd one class as an undergraduate, which was the biology lab class. And the story behind that is I took the class, I don't remember, either in my first or second year, probably early in my second year. And one of the class was taught by several different people, but one of the people who taught the class was Nancy Hopkins. And she was, at that time, she was just a starting assistant professor. So I think she was early in her career. And early in the class, we had to write a lab report for something. I guess I must have taken this the end of my first year. And I had no idea how to write a lab report. I didn't know what the format was, and you know, I didn't know that you were supposed to lay out a hypothesis, what you design, you know, how the experiment was designed, what your results were, and what you conclude. I didn't know any of that. And I got back an evaluation from submitting this report from Nancy. It was like one of these see me things. It was just so bad. And she said, you know, Greg, this is not how you do a lab report. And she went back over it. But then later on, when I was a senior, I must have improved significantly during the class. She asked me back to TA the class.
Wow, that's really cool.
Yeah, so I did TA.
Yeah. And Nancy was teaching biology.
She was a new assistant professor in the Cancer Center in biology. There's a great book that just came out, which you should read. It's called The Exceptions. It's about all the challenges she faced as a woman in the MIT biology department at that time.
Wow, I must read that.
It's called The Exceptions. It just came out recently, I think in this year, in the last 6 months.
And then how did you then decide to go pursue a PhD? Because that's a huge decision. I remember when.
Yeah, when I was at MIT, I mean, you know, by the time I finished MIT, I mean, other people who had been big influences on me were the guy who was my undergraduate advisor. He was a bacterial geneticist named Boris Magasanik. And another relatively young guy who was a geneticist at the time, whose name I'm forgetting now, famous geneticist, David Botstein.
Oh, David Botstein.
David Botstein taught my undergraduate genetics class. And I actually also got invited to, I didn't TA the class. In the TAs, you also had, it was a big class, you had section leaders. So I was a section leader in that class too. And they both said, you know, I think it's a good idea for you to do this. And I really wanted to do basic research. I wasn't really successful doing a lot of experiments when I was an undergraduate. I tried to do a whole bunch of different things, and I sort of played around a lot, but I was actually really concerned with making sure I could graduate in time. I actually graduated a semester early, even though I was majoring in
Not surprised.
in two disciplines, because it was just expensive, and you know, I have two siblings. By that time, they were both in college, so it was, and my father had retired by that time, so it was kind of a financial drain. So I actually graduated a semester early.
And then
I was, you know, was looking around for graduate programs, and you know, I I applied to several different places. I really wanted to go someplace where people were working in new areas in biology. I didn't want to do conventional stuff. It was clear I didn't want to do what I would call old school biochemistry, although I ended up doing most of my PhD as doing biochemistry. And so I applied to various places. I eventually ended up going to Caltech for a variety of reasons. When I came out to Southern California, it was about this time of year. It was February. The weather in the East Coast was was nightmarish, and it was beautiful in Southern California. And it's a different era. At that time, the graduate program at Caltech was not very big in biology. I think they probably admitted something like 10 students a year, distributed all over biology.
Wow.
And, I was pretty heavily recruited. I was just impressed by the whole process. I remember the main person who was handling, the graduate recruitments at that time was somebody who subsequently went on to a fantastically successful career, a guy named Lee Hood.
Yes.
And I remember Lee calling me up at 7:30 in the morning, East Coast time.
Wow.
And so I realized it was like the middle of the night here on the West Coast, and he was so I ended up being at Caltech, which at the time was a great place to be a graduate student. So graduate students were very heavily involved in the research. They were there at the time. They were treated with a great deal of respect, and it was a great place to be a graduate student.
No, I know that, circadian rhythm, the molecular genetics of circadian rhythm started Caltech.
That's right. That's right. So when I was there, Ron Konopka,
who was a grad student,
who had been a grad student with Seymour and published the famous paper, this famous PNAS paper, one of the most influential papers ever. He was already an assistant professor at Caltech. It was actually, well, that's completely another different story. It's interesting how Ron had actually brought that project to Seymour's lab.
Yeah.
So Seymour was a fantastic scientist. And a very, very kind human being, one of my scientific heroes. He was interested in the general idea as to whether or not you could use genetics to study behavior, whether you could isolate single genes that will control various aspects of behavior. But the idea of using Drosophila biology to get at genes involved in circadian biology was really brought to the lab by Ron Konopka as a student. He was a graduate student with Seymour. And Ron knew that this process of eclosion during the larval life cycle of Drosophila followed a very clear circadian cycle, and so it was actually quite easy to assay that.
Yeah.
And it was really Ron who brought that there.
Yeah. But that was a beautiful piece of work in PNAS.
It's an amazing paper. It was an amazing paper because in that single paper, they isolated this gene per, mutations in this gene per, and in one paper, they got a mutation that shortened the circadian period. They got another paper that lengthened the circadian, and another mutant that, it's all in one paper, and another mutant that destroyed the period, that made the eclosion aperiodic. And so it was really quite remarkable.
Yeah, and actually, there's some little connection between that paper and myself in the sense, that paper was published in PNAS on the day I was born.
Yeah.
The 2nd July 1971.
Wow.
So that's it. But that paper is the inspiration behind this Pulitzer Prize-winning book, Time, Love, and Memory. So that's another great book it's mostly on Seymour's work on Drosophila, using Drosophila as a model to figure out circadian rhythm and few other things. Okay, so then what is your project in Caltech?
I actually started off in Lee's lab, but very quickly I moved to neurobiology lab. There were a lot of well, there weren't a lot. There were a few people at Caltech who were starting to apply cellular and molecular techniques to the nervous system. This was a completely different era. This is like, now we're talking the late 1970s. So you still would have been a young child. People were just starting to do this. And so I eventually worked with a guy named Jeremy Brox, who had been a postdoc with Martin Raff in London. And people were just developing techniques at that time, new techniques, which now seem so incredibly trivial, but back then they were a big deal. And this was using antibodies to identify particular cell types in the nervous system and using secondary antibodies that were fluorescent. So this was like the big technique of the day, to make these glowing cells that were red or green or something like that. And people would just thought this was the most amazing thing. It's, I've seen various techniques, come and go since then. The current techniques that everybody's kind of blown away by now, I think in 10 years, everybody will say, yeah, this is either outdated or we all take it for granted now, right?
Yeah, so, that reminds me, I don't know whether you were at that conference in Salk, Paul Schimmel from Scripps. The title of his talk, or I guess the theme of his talk, was "We are living in the dark ages of medicine." Because whatever you feel is awesome today, in 5 or 6 years, we'll feel like, oh, that's so obsolete. I can't believe that we are doing science that way.
And particularly for medicine. I mean, we're still, and we very much are in the dark ages of human biology and human medicine. There's no question about that. Paul Schimmel, by the way, was at MIT when I was at MIT. He had not yet moved to Scripps. So I worked with Jeremy. I worked on, Jeremy had an article in Nature, was actually on the cover of Nature, where he'd had an antibody to a particular cell type in the peripheral nervous system, the Schwann cell, which is derived from the neural crest, which forms myelin around all the peripheral nerves. And so he had identified an antibody which allowed you to identify Schwann cells and to purify them. And with Raff, he had also started to develop culture techniques where you could grow the cells. And you could purify the cells from peripheral nerve, from mammalian peripheral nerve, either mouse or rat. We did mostly from rat, and then grow those cells in culture.
So these are the cells that wrap around the axons, and
these are the cells that wrap around axons, make this thing called the myelin sheath around axons, which is actually the insulation around peripheral axons that allows nerves to conduct much, much more rapidly. They're the cousins of the equivalent cell in the brain called an oligodendrocyte, which does that around cells in central nervous system. So the ability to form myelin sort of tracks basically with vertebrate evolution, and it's essential to the evolution of nervous systems, because without that, the only way you can get rapidly conducting axons is to make them bigger, is to make them enormously large. And our brains don't have enough space to make all the axons big. So myelination is a key component in the evolution of nervous systems. And we had the ability, at the time, we were one of the few labs in the world trying to do this, trying to isolate these cells, grow them in culture. And my PhD thesis was actually to isolate a growth factor for Schwann cells. We knew that if you made extracts from brain or pituitary under the right conditions, you could extract an activity which was a mitogen for these cells, which drove their division, and that allowed you to expand the populations greatly.
So you were actually extracting this factor from sheep brain or cow?
We were mostly doing it from cow pituitaries. So, we were following on the kind of the footsteps of peptide biology here at the Salk, and the work of Roger Guillemin and Wiley Vale. We didn't know this at the time, but we were basically using some of their early protocols and steps. So my PhD thesis was to purify this protein, which I eventually did. And I showed we called it glial growth factor. It subsequently turned out that this protein is the same protein that other people were trying to purify from other bioactivities. It's called neuroregulin-1.
Oh, okay.
It's a protein which acts as a ligand for a family of receptor tyrosine kinases, which my lab subsequently worked on a lot. So, but at the time, it was, we'd purified the protein. When I finished my sequence, it was right about the time you could sequence proteins from small amounts. Lee Hood was actually very important in this. But I had actually finished my PhD thesis by that time and had moved on to my postdocs. I didn't know what the sequence was, but it turned out subsequently to be neuroregulin-1.
Neuroregulin. Yeah, so in those days, the PhD thesis used to be cloning a gene.
Well, no, this is actually even before cloning a gene. So by the time I was doing my thesis, it was just the time when libraries, either genomic or cDNA libraries, were the vectors, the bacterial and the phage vectors for generating those libraries were being developed. So when I went to Caltech, during the time that I was a graduate student at Caltech, the kind of hot young star in the field for generating those cloning vectors was at Caltech. That was Tom Maniatis. So he was an assistant. Tom Maniatis was an assistant professor at Cal, came as an assistant professor at the time. And his big thing was he was making these lambda-phage vectors that allowed you to, with DNA from various sources, it could be any source, human thing, you could package those DNAs up and make a library which had a representation of all of the genome. So that was the big thing at the time. And when I finished my PhD, it was just about the time that people started doing that for so-called cDNAs, that is to say DNA transcripts of the mRNA. So that was just, those libraries were just being developed. In fact, my first cloning paper, which I published as a postdoc, the particular vector I used to make that library and to clone that cDNA was still unpublished.
Wow.
So that was a little bit difficult. I remember getting the paper posted because we had to actually, there was a bunch of description of the vector and we had to reference a whole bunch of unpublished stuff because it hadn't been published yet.
So after cloning, or not cloning, after identifying neuroregulin, which you didn't know at that time, then you moved to finding the receptor.
No, that was even later. So what I did for my postdoc, so this is still related to Schwann cells.
Yeah.
So what I did for my postdoc related to the myelin that the Schwann cells made. Yeah. So we had known actually from Jeremy's lab, from work that Jeremy had done, there were a set of proteins that defined peripheral myelin. And the major one of these proteins was a protein called P0, protein 0, or myelin. It's now called myelin protein 0.
Why protein 0? Because I have never heard of 0.
I don't know why they started with 0. They just ran these isolated myelin proteins out on a gel and numbered them. 0, 1, 2, 3, 4.
I see.
And they all had different. And so P0 was the major protein. It accounts for about 50% of the protein in peripheral myelin.
Wow.
And it's only in peripheral myelin. It's not in central myelin. In the CNS, the oligodendrocytes make a different protein. And it's remarkably specific to these cells. I mean, at the time, this was an issue in gene expression. It's like, how do you get certain genes that are only expressed in some cells? And this particular protein was remarkable. It couldn't be detected anywhere else other than myelin, and there was tons of it, and its expression in the Schwann cells was tied to their stopping dividing and making this and making the myelin sheath. So it was coupled to myelination. And I wanted to learn how to do molecular biology, how to make libraries, how to clone genes out of libraries. I thought this should be pretty easy.
Because 50% of the protein is...
50% of the protein is this thing. So the problem was, it was still very primitive days. So the only thing I had was an antibody.
Oh.
And expression libraries hadn't been developed yet. So we only had... So what I had to do was to take... I made a library from peripheral nerve during the time that myelination was really active. So there should be a lot of this mRNA. And then I screened that library for genes that were expressed in peripheral nerve but not in brain. It was a really primitive differential screen.
Differential expression screen.
Differential expression screen. And so then I took those clones that were positive in that screen, and I used the clones, this was just a subset of the clones initially, to select mRNA that was expressed in peripheral nerve by hybridization, then to elute that mRNA off, translate it in vitro using rabbit reticulocytes. It was really a brutal cloning. Rabbit reticulocyte lysates, and then to immunoprecipitate those in vitro translated products with the antibody that I had.
Yeah.
And that's how I identified the thing. So
Wow.
yeah, it was.
So this was from mouse or which?
This was initially it was from rats because the rat cells were much easier to grow.
Yeah.
But then it went on, and so I cloned that.
What is that protein?
MPZ, myelin protein. MPZ. So for people, MPZ it turns out there are many mutations in MPZ that lead to peripheral neuropathies, which neurologists can diagnose in patients because they have an altered gait, they have problems moving their limbs, and the reason there are so many mutations in the gene is because a lot of these mutations the effects of the mutation don't affect the development of the human embryo. So the embryo is born, actually they can move depending on the mutation, they can move pretty well for a while. And so the things are subtle deficits, with pain in peripheral nerves when people get a lot older. So I think the protein is relatively small. It's only about 30 kilodaltons. I can't remember how many amino acids are on the protein, but I should because I was the person who cloned it. But something like half of them are known to be mutations now. So if you go into
300 amino acids or so.
Yeah,
roughly.
and a lot of them are mutated. So
that's
and it turns out that kind of relates. But I was still working on Schwann cells.
Yeah. And then how did you go back to neuroglin and?
Well, early when I came here to the Salk Institute, again, it was another sort of technical.
But your postdoc was back in where?
My postdoc was with Richard Axel at
Nobel Prize for.
Yeah, who much later got a Nobel Prize let's see, how much later? About 20 years later, got a Nobel Prize for working with Linda Buck for identifying olfactory receptor genes. But at the time that I was there, Richard's lab was mainly a cloning lab. So there were many people there just trying to clone interesting genes, and that spanned all kinds of biology. So David Julius, who subsequently also went on to win a Nobel Prize, he wanted to clone the receptor for serotonin in the nervous system because those receptors were not identified. He did that. And my good friend, so the other good friend I have from that era, other than Linda, is Dan Littman, who's an immunologist.
Yeah, Dan Littman.
Dan was really interested in genes that distinguish T cell subsets. And so while he was with Richard, ended up cloning the genes that distinguish the two major T cell subsets in the immune system, CD4 positive cells and CD8 positive cells. So he cloned both of those genes. So it was a cloning lab. And when I came here, I was still a bit into that mode.
But how did you decide to come back to Salk? This Southern California pool? Or?
There was a big Southern California pool. I knew a lot of folks who were here in San Diego, mostly at UCSD, not so much here at the Salk, but there were two neurobiologists here at the time who were applying molecular biology to the nervous system. Again, this was kind of a new thing.
This was in 80s?
This was in the 80s, and this was Steve Heinemann and Jim Patrick. And so, let's see, this would have been about the mid-80s, about 1985 or 86. And they were interested in cloning neurotransmitter receptors. They were very interested in cloning the receptor for the neurotransmitter acetylcholine. That's what their labs worked on at the time. Steve, of course, they were ultimately successful in doing that. Steve went on to clone all of the receptors for the amino acid glutamate, which is the major excitatory amino acid in the nervous system, in the brain. But they were working on that at the time. That's what really attracted me. There were a lot of people trying to do molecular biology. And I continued to work on the Schwann cell aspect of things for quite some time. But I got very interested in, work that my colleague Tony Hunter had been doing on tyrosine phosphorylation and receptor tyrosine kinases.
Yeah.
So I had a postdoc who came to the lab at the time. He said, "Why don't we work on receptor tyrosine kinases in Schwann cells in peripheral nerve?" I said, "I think that's a great idea." So,
but you are just starting assistant professor.
I was a starting assistant professor.
Most starting assistant professors are very conservative. They would like to get their own paper.
Well, I published some more stuff on. I published some more stuff on myelin and on Schwann cells.
I see. So
we published more stuff on that, and those were my initial papers.
Yeah.
But this postdoc I want to do was a guy named Kerry Lai. And Kerry was an incredibly smart guy, and so this is before genomes had been sequenced. We didn't know how many receptor tyrosine kinases or tyrosine kinases in general there were. But several had been identified and cloned, so we had their sequences. And this technique of polymerase chain reaction, or PCR, had just been developed.
Yeah.
And so he said we could do degenerate PCR based on the known sequences from a tissue source, and we could see how many we can clone. And of course, a lot of people had this same idea.
Yeah.
But so a lot of people were trying to do this. But Kerry, in talking to Tony Hunter and to people in his lab, they pointed out the fact that the sequence that we were trying to amplify between different tyrosine kinases was essentially invariant in length. It was always, it was like maybe one amino acid difference, but it was generally they were all exactly the same length. And the problem that people had at that time is they would run their degenerate PCR reactions. And then they'd take what they got out of the PCR and they run it on an agarose gel. They'd cut a band out of the agarose gel. They'd clone that band and they'd sequence all their clones, right?
Yeah.
The problem with doing that was that there was the degenerate primers that you use would amplify a lot of junk.
Yeah.
And so in that band that you cut out, even though it looked like it was the right size, you would go through the sequencing thing, and most of the stuff you would sequence was not a kinase. It was just garbage. It was actin or something else.
Those days, sequencing was very expensive too.
Sequencing and labor intensive. We still did manual sequencing in the lab.
Wow, those old gels.
Yeah, we ran the gels ourselves. Yeah, and there was no sequencing companies. We did it all in our labs. And so these labs were just spending an inordinate amount of time to sequence junk.
And a lot of radioactivity.
A lot of radioactivity. So they would publish these papers where they would sequence, and then we'd come out like one new kinase. So Kerry, based on his conversations with Tony, it was like a trivial thing. But instead of running the sequencing reaction out on an agarose gel, he ran it out on a polyacrylamide gel. So he would have much, much, much finer resolution in the size of the band. So then he only cut out the band that was exactly the right length from the polyacrylamide gel and cloned that. And when he sequenced that, everything he sequenced was a kinase.
Wow.
So we now...
Did you sequence?
We didn't sequence that. The first paper that we've only published one of this sequencing papers. And he just did it from peripheral nerve, from sciatic nerve, you know, the main peripheral nerve of the body. And I should have I'm BSing here, I can't remember the exact number. We now know from sequencing the genome that there are 58 receptor tyrosine kinases in mammalian genomes. So the maximum number you could clone was 58. And I think in this one paper, Kerry cloned about half of them, like 20-some. And of those 20-some kinases, 11 were completely novel and new. They were unknown. So like, in one paper, he published we would learn stuff like this later when we're sequencing the genome. You uncover all these genes, it's like, but in one PCR paper, he cloned 11 novel kinases.
Oh, this is amazing because in those days, just cloning one gene
was a
big gold mine, hitting a gold mine.
Yeah, exactly. He cloned
11.
He cloned 11. Now, of course, what he cloned out were just short fragments of the kinase. But then he did, he did a multiple sequence alignment of these things. And he said he segregated them into different families. This is when the families were still being even named. He cloned like 4 new EPH receptors. He cloned another neuregulin-1 receptor, which we subsequently went on to work on. He cloned 2 new families, identified 2 new families that are called DDR and the TAM families, which my lab then went on and studied for decades. But it all came out of one paper. And it was all because of thinking about, from first principles, about how to do the PCR reaction correctly.
He was a postdoc or grad student?
He was a postdoc.
Yeah, that's also one of the magic sauce of the Salk Institute because...
Talk to your colleagues, yeah.
Awesome postdoc or grad student or trainee or technician comes in and then talks to neighboring lab and improvises on one simple technique. Most of the time, it just boils down to doing the experiment slightly differently from...
Yeah, this was a trip. I remember there was this excellent scientist in Australia named Andrew Wilkes who was trying to do exactly the same thing.
Yeah.
And, Carey's paper just blew all these labs out of the water. I mean, it was really...
So just going from agarose gel to polyacrylamide gel. Yeah. So then how did you then move towards TAM receptor?
Well, we applied another new technique at the time, which was another new technique. So then we cloned full-length cDNAs for these things. We could see what the structures of these receptors look like. And then, you were confronted for many of them, if it was a completely new gene, we used a new technique, which was to make mouse mutants for the gene, to make mouse knockouts for the gene. Yeah. Again, this was like in the 90s, the early 90s, this ability to inactivate genes in the mouse and to make mice that lacked a particular gene, that was kind of a new thing.
Yeah.
And so we just started doing that. And we sort of worked our way through, we had a small lab at the time. We had to decide which genes were most important. So we initially did, one of the initial genes we did was for this additional neuregulin receptor. So these receptors had been intensively studied, the neuregulin receptors, in the context of cancer, because one of them, ERB2, or in humans, HER2, is a gene that's commonly mutated or amplified in human breast cancer, right? Yeah. And neuregulin-1 is actually the ligand for that thing. In addition, so we had cloned a new neuregulin receptor. It was homologous to HER2 and HER3. Mm-hmm. It was called HER4. We called it ERB4 in the mouse. Yeah. And so we knocked the gene out of the mouse and asked what the consequence was. And so we did, and then we published a bunch of stuff on that. Well, not a bunch of stuff. We published several papers on that receptor. Carey subsequently went on to work on it in his lab. The really satisfying part about that analysis was when we made the mouse knockout, we first of all showed there was an essential effect of this receptor in development of the heart. So
this is kind of interesting. So you cloned the gene from sciatic nerve, and then you realized that at that time it was known HER2 was already cloned? No.
Well, HER2 had been cloned.
But so you immediately knew that it's...
We knew it was related. It was structurally related to HER2. It looked very similar. Yeah. Its kinase was very much like HER2. It's a little bit different. I mean, there are important details in the binding. All these receptors work as dimers, and even though neuregulin-1 had been cloned as a putative binder of HER2, it actually doesn't bind HER2 directly. It binds another protein called HER3, And HER3 forms a heterodimer with HER2. Yeah. And that is actually the signaling complex that works in the mammary epithelium. That's what the complex is in breast cancer. HER4 does bind neuregulin directly, and it can form dimers with HER2, but it can also form what's called a homodimer, 2 molecules of ErbB4, and that actually signals in the heart.
Wow.
And we didn't know that until we made the knockout. So when you made the knockouts at that time, the best phenotype you could have was a dead mouse. Yeah. Because they played some essential role in mouse development. And we had a dead mouse. It died. It died in the middle of embryogenesis, and so we had to figure out why it died. Yeah. And it turned out that that's because there's a signaling interaction between the muscle cells in the heart and the endothelium of the heart, the embryonic heart. Mm-hmm. The endothelium has the ligand, neuregulin-1. The cardiac muscle cells have the receptors, mm-hmm. and that the muscle cells in the middle of embryogenesis have to undergo this morphogenetic change called trabeculation, which thickens the wall of the heart and allows the embryonic heart to pump blood through the. Oh, I see. So you can see that if you've ever seen a section of even of an adult heart. Mm-hmm. And so that was the phenotype that we had at that time. But to get back to the satisfying part is, subsequently, the postdoc who made that knockout is a guy named Martin Gossman. He specifically rescued the heart phenotype and then asked what happens in the rest of the embryo if the mouse embryo survives, because it has an immature heart. And then it has all of these Schwann cell phenotypes. So there are basically no Schwann cells in peripheral nerves. You need that neuregulin signaling system to have Schwann cell proliferation. Since there's no Schwann cells, there's no myelin, so you have all of these major phenotypes, and that related back to what I had studied as a graduate student, because the growth factor I had purified, neuregulin-1, signals through the ErbB system.
So how many years was that from?
Well, it wasn't. That was about, let's see, I'm trying to think. 10? By the time you made that rescue, more like about 15.
15 years, yeah. So that's what I kind of gathered, that almost you work on one big problem, it takes 10 to 15 years to.
Well, in part it takes 10 to 15 years, but it also takes time for the techniques that you need to do the analysis to advance. So, what Martin had to do was to transgenically express the ErbB receptor system, HER4 and the other receptors, specifically in the heart, using a heart-specific promoter. So, you're required on the development of those techniques in order for you to be able to do that.
But it's also another thing, in those days when people are cloning new genes, since the genes were expressed in different stages of development or in different organs. Right. You have to be a little smart to figure out where the phenotype is and then go back to detect.
So if you make a mouse knockout, as I said, you would only see the first place that it has an essential role, because then the embryo would die. If it subsequently did something essential later, you had to do more fancy experiments. Yeah.
So that's how you went back to this one cell, and then how did you come to TAM receptor?
Well, the TAMs were the best part of the story, because they were
let's say a little bit about TAMs.
So the TAMs were a completely new family. So they didn't, unlike a lot of the other stuff we cloned, they didn't look like a receptor, a known receptor. So there's something called fibroblast growth factors, which were originally identified here at the Salk. So FGF receptors had been cloned. So we cloned some new FGF receptors. We cloned some new EPH receptors. We did a lot of work on those too. But those were, when you looked at their structures, they looked like the identified things, whereas the TAMs looked completely different. And
so they also came out of this.
They also came out of the original screen.
Oh, okay.
And so, eventually we did the same thing through those receptors. We made single knockouts, and other labs had made in the intervening times had started to make single knockouts of the TAM receptors.
Who named them?
I named them because at the time I could name them as TAM, just for the 3 receptors that subsequently. Yeah, it's a little bit complicated. So
Let's go back to the name.
So the naming, we originally called all of our clones tyros. They were
Tyros.
tyro 1, 2, 3, 4, 5, 6, 7, and we named them all tyros. And then the way naming conventions go is I'm pretty sure, the first person to clone a full-length cDNA,
Yeah. Guess it.
their name gets to be accepted. So the TAM family was originally, in our nomenclature, was called tyro 3, tyro 7, tyro 12.
Yes.
Right? Tyro 3, 7, 12. In our first papers, that's how we referred to the family. And then we cloned a full-length tyro 3 cDNA. So we got to
Tyro 3.
tyro 3. The tyro 3 was our accepted. So the naming gurus, I don't know where they are. Somebody says, okay, it's tyro 3. But we didn't clone full-length cDNAs for tyro 7 and tyro 12. Other labs cloned them. And a lot of people were working on these. The first one that cloned a full-length cDNA was called tyro 7 Axel. And the first person to clone a full-length cDNA for tyro 12 called their receptor MER, M-E-R.
Yeah.
And so, but Axel and MER also had a zillion other names. And so the literature, and this was true for many genes at the time.
Yeah.
Different labs, and they had their own labs, says, no, we're not going to use your name. So they were all these complicated names, and eventually it just got to be, so Axel was also called UFO, it was called ARC, it had about 6 different names. And so I said, let's just call them, since Axel and MER are now the acceptance agent and tyro 3 is accepted, let's just call the family TAM for tyro 3, Axel, and MER. We'll call it the TAM family. So I was the one who didn't get that just because the literature had started to become impossible and people weren't sure what was what. So
I see.
So I coined it.
Okay. So then these TAMs, they started from swan cell, but they're expressed
They're widely expressed.
everywhere.
They're widely expressed in the family. The TAM family, tyro 3 is quite distinct from its expression, and Axel and MER tend to be in the same cell. So we made knockouts of all of them. And other labs had made knockouts of Axel and MER. Oops, you're not communicating now. And then other labs had made knockouts of Axel and MER. And so we started doing this. And the initial knockouts, it was kind of a disaster for the graduate students who made the knockouts because the mice weren't dead.
Mice weren't dead.
The mice were not dead. And I can still remember the graduate student at the time who made a tyro 3. He said, my mouse has no phenotype, by which he meant it wasn't dead. And so we and several other colleagues, we eventually got all these mutants into my lab, and we started making multiple mutant combinations. We eventually made a triple knockout mouse that had no TAM receptors. It didn't have any tyro 3, it didn't have any Axel, it didn't have
So none of the single mutant had any acceptable
It
phenotype.
turns out they had really interesting phenotypes,
But initially,
initially no. So the first paper we published was actually a triple knockout paper. And again, this was a technical tour de force at the time.
I remember, I think it was in 2099.
It was right around 2000. It might have been 1999.
Yeah.
And if it wasn't the first mammalian triple knockout, it was one of the first. I mean, because this was like, it was a pain in the ass at the time. It was a technical tour de force. And the triple knockout, when it was born, it was indistinguishable from wild type.
Really? So
So that's
one out of 64 will be a triple knockout.
Exactly. Exactly. There's all that. There's all of that problem. There's all the problems of just the arithmetic of doing mouse breeding. It's horrible. But the triple knockout, the paper we published was a paper in Nature. It was a prominently published paper. The triple knockout males did have one really interesting phenotype, which is that as adults, they were infertile.
Okay.
And when we started looking at it, it was really quite a clear, dramatic phenotype. There was essentially no cells, no in the germ lineage of the testis, of the seminiferous tubules of the testis. Basically, it was completely blown out and destroyed. And so that's why the males were infertile, because there was no spermatogonia, there were no mature sperm, there were no cells at any stage of meiosis. They had almost all died, and it really looked like a wasteland. I mean, most of the tissue was just destroyed.
No, this is also another interesting point, because there are many mutants that also have reproductive defect, but very few people actually chase down to see what is the problem. And you, coming from Schwann cell and neuroscience, actually chase down to see what is the defect.
Right. So, the reason for that subsequently turned out, we actually didn't quite realize this in the Nature paper at the time, but it turned out to be one of the main things that the TAM system does. In particular, well, all the TAM receptors in the testis, but in other places, it's mainly axil and MER, is the TAM system is required for the recognition and the phagocytic eating of dead apoptotic cells in the body, right? So a lot of people don't appreciate this, but in normal human life, as you're talking to me now, something like 2 to 3 million cells are dying in your body per second.
Yeah.
And they have to be recognized as a part of normal cellular turnover, and they have to be recognized, and they have to be phagocytically eaten by professional phagocytes. In the immune system, these are mainly macrophages, but in the testis, it turns out there's a somatic cell called the Sertoli cell, which does this. And during meiosis, to generate mature sperm, the cells go through meiosis, but most of the cells never make it to mature sperm. So going from a spermatogonium through meiosis to mature sperm, something like 80% of the population normally dies.
Really? I didn't know that.
This is a normal thing. What? It turns out that's true in all places in the body where you're making a lot of new cells throughout life. Most of those cells die.
Yeah,
like an adult neurogenesis in the brain. Most of those cells never become neurons. They die.
I see. Yeah.
And so you need cells to see those dead cells and to eat them, to turn over their components, to recycle the materials. And the TAM receptors are actually critical for that. So they're expressed by the Sertoli cells. The Sertoli cells that don't have the TAM receptors, they don't see the dead cells. The dead cells become necrotic. They spill their guts all over the tissue. They poison every cell. They kill every cell in the tissue. So that was that phenotype.
I see.
But it subsequently turned out there's a similar. So this is something you can probably relate to. So normally in the retina, for about an hour a day after you wake up, immediately after light enters your retina, after you open your eyes and you've been sleeping all night, there's a process of phagocytosis that goes on with a phagocytic cell in the retina called the retinal pigment epithelial cell, or RPE cells. And it turns out that during the process of phototransduction, of photons entering a photoreceptor, they generate toxic oxidated lipids and proteins as a result of receiving all these photons into the cell. And those toxic products will eventually kill the photoreceptor if they're not gotten rid of. And so what the photoreceptor does is, if light's entering here, and the photoreceptor outer segment's up here, which has all the rhodopsin, which allows you, your kinds of proteins you're interested in, that allows you to see light. It puts fresh membrane at the base of the outer segment, and it pushes the old
to the top.
To the top. And at the top, this phagocyte sits and just pinches off a part of the cell. It doesn't eat the whole cell, because you can't replace the cell. It just prunes the end that has all the toxic stuff. And it turns out you need TAM receptors to do this. So even though the graduate student for the MER knockout said, my mouse has no phenotype, the mouse was actually blind.
Okay.
And you will know this more than most people. For a mouse that's sitting in a cage, you're not going to know that the mouse is blind unless you do a test. right?
Yeah.
Because the mouse can still find food, it can still find a mate to have sex with, it can still live its normal life,
Yeah.
being completely blind.
Yeah.
Right? And so I remember years later, it took us at least a year before we happened to randomly section through the eye.
But how did you get to the eye? I mean, I'm always curious about.
It was totally fortuitous. I mean,
No, I don't know. I don't think it's fortuitous.
we were looking for phenotypes.
Yeah.
We were looking for phenotypes in the brain, so we had cut sections through the brain which contained the eye. And the postdoc at the time said, I don't think this retina looks quite right.
No, that's the point.
And I looked and I said, yeah, it's missing half the cells.
Yeah.
Because the photoreceptors are a big, thick chunk of the retina. I mean, if they're gone, I mean, it's easy to see.
Yeah.
And I said to the graduate student, you said your mouse has no phenotype. I bet if you asked the mouse, it would say, yeah, I can't see a thing. I have a phenotype. So that was...
No, that's the thing. Like, you have to look. Means you looked for the triple mutant, you looked into the spermatogenesis, and then here you're looking at the eye, and you started from the sciatic nerve. So this is like the pure curiosity of a scientist.
I mean, the spermatogenesis was, first of all, it only develops as adults. So if you look at the mouse testis, the triple knockout testis, before the mouse is sexually mature, before it starts to try to have the spermatogonia go through meiosis, it looks perfectly normal.
Nice.
It's all the cells have developed perfectly fine. They're set up. It looks beautiful, just like wild type.
Yeah.
But once the cells start to go through meiosis and the cells start to die and they start to pop, then you see the phenotype. And it's the same thing for the retinal degeneration. A mouse when it's born has its eyes closed.
Yeah.
And it gets, there's still some light coming in, but it's nothing like when the mouse opens. It's only when the mouse opens its eyes and there's this massive influx of photons into the retina, which in a mouse, you will know better than I think occurs somewhere after the second postnatal week.
Yeah. 14 days.
Well, about 14 days, they stay
open,
and then you start to see this phenotype, okay? And subsequently, it turns out that they're human beings. If it's the same physiology happens in you and I. I mean, you have to homozygose the mutations, so they're rare mutations. But people who have complete loss of function in MER have problems seeing things, because a lot of the photoreceptors are dying.
Yeah.
So it's the same thing in human beings, but you don't follow this until in the immune system it was the same thing. The mouse is like running around. It's kind of fine. It looks a little sick, but then when if you sacrifice a mouse and look at its organs, its spleen, by the time an axil MER double mutant, for example, by the time it's about a couple of months old, its spleen is 10 times the size of a wild type spleen.
So, because all the dead cells are still there.
Well, so your spleen is one place where it's probably the major site in your body where you have to turn over dead cells. There's two populations. One is erythrocytes. Yeah. So erythrocytes only live in your body for about a month and a half, I think, in humans, or maybe 2 months. So they're turned over so every year there's a huge turnover of those cells. And neutrophils in the immune system have a very short half-life, I think 10 days. And so they're constantly being made. And so in the TAM mutants, those cells are just piling up at the spleen. You have terrible, and you eventually develop bad autoimmune disease. So that paper in immunology, which again was prominently published, it was on the cover of my very first paper that I ever published in immunology, was in a special issue of Science devoted to immunology.
Wow.
And it was the kind of paper you could publish back then, you can't publish now. So that paper was Liu and Lemke. It was done by Qingxian Liu, who was a postdoc in Lemke. It's a two-author paper.
Two-author paper.
My paper cloning P0 as a postdoc was Lemke and Axel was a two-author paper. Yeah. Those kind of papers don't exist anymore. But
you used to work in the lab when you were the PI and then the postdoc.
Yeah, although by the time Qingxian started doing the immunology, I don't think I did any experiments.
Look into the slides?
Oh, yes, definitely.
Yeah. I mean, these days...
Definitely. I mean...
You may not be sectioning the
No, I wasn't cutting the sections, but I was definitely, but I didn't know the immunology that I had learned was immunology that I had learned when I was in graduate school. I can still remember Ching Shen coming into the lab and saying, because we'd started to look around and there were these pools of activated lymphocytes, of T cells and B cells, basically in every tissue in the body, which is very abnormal, right?
Yeah.
We would cut a section of muscle and there'd be this big colony of T cells there. And
But he must be standing for them, right?
he was standing for them, but there was enough of them. It was pretty easy. If you looked, it was pretty easy to see.
Yeah.
And he came into my office one time and he said, I've started to look, and he says, I know we see all these pile up of dead cells and then T cells and B cells in all these tissues, but the receptors aren't expressed in lymphocytes. He said, they're expressed in monocytes and monocyte derivatives. I said, that's very interesting, Ching-Shen, thanks. And after he left my lab, I pulled down the immunology textbook.
Yeah.
I looked in the index for the chapter on monocytes and I started reading about it so that I would know what macrophages and monocytes derivatives were. That's how much immunology I knew. But when we finally got around to publishing it, we were on the cover of a special issue of Science devoted to immunology. That was my introduction.
Yeah. So that was the triple knockout, or
I'm trying to remember. That initial analysis was in the triple knockouts, the straight triple knockouts. But it subsequently turned out that most of that phenotype of the immune system is due to knockouts of AXL and MER. And depending on the cells you look at, MER is the most important cells. And those, again, this is at the time, these were what's called germline knockouts, so they were conventional knockouts. The gene was knocked down in every cell in the body. It was before, again, there was another technique that had to develop where you could make where there were cell-specific Cree drivers, and you could make your allele have LOXP sites that would allow you to inactivate it in specific cell types. But at the time, we didn't have that technology. I mean, that was developed actually probably more than six or 7 years later. The very first ones of those were published. Because that paper I was telling you about, that was published in 2000 or 2001, I think.
I see. So what are the ligands for TAM?
So there are two ligands, two proteins. That was another thing. The TAMs, to go back to the origins, and when they were what was called orphan receptors. So they didn't look like conventional. It didn't look like an NRB receptor, so you could say the ligand was going to look like neuregulin. It didn't look like an FGF receptor, so you could say the ligand was going to look like FGF. We had no idea what the ligands were. So that we really didn't do much in my lab. We made cells that expressed these various receptors, cell lines that didn't normally have it, that had the receptors. And we sent no, we didn't do this. It was just cell lines with the first receptors. We sent these to someone I had known from my postdoc named George Yankopoulos, who went on to found probably one of the most successful biotech companies ever, Regeneron.
Regeneron, okay.
I think George is commonly referred to as the first biotech-generated billionaire.
Oh.
So he was one of the very first. He started Regeneron.
Yeah.
And so he had a big armamentarium of people who could basically use the cell lines that we sent him to fish out the ligands. So it was primarily George who identified the ligands. There were labs at Amgen and Genentech working on identifying these ligands too, but I think George was the first. And there are two proteins. They're called Gas6, which is only a ligand for the TAM receptors, which actually binds to all three receptors. All this took a while to work out. And then another protein called Protein S, which has another function. It's an anticoagulant in the blood coagulation cascade.
Yeah.
And George cloned both of those. Protein S is a ligand for two of the TAM receptors, for tyrosine and for MER, but not for AXL. So we had to find the ligands too, and make mutants for the ligands and all that.
Yeah, and then how did you connect it back to the brain? Because
Well, there's a part of the brain. I mean, we got again, this is a long story. You sort of have to just follow how things go. There's a part of the brain which our colleague Rusty Gage here has worked on for a long time, where there is continuing generation of new neurons from precursor cells throughout. It's much more common early in life, but it persists throughout life. And in these neurogenic areas, it was known from other analysis that it was like the testis in the sense that most of the cells that start to try to become neurons from the progenitor cells die. And so they have to be cleared. By this time, we had already known that the TAMs were among the most important clearance systems for macrophages throughout the body. And in the brain, you have a specialized set of tissue macrophages called microglia.
Yeah.
They're basically the brain's dedicated tissue macrophages. And we knew that they probably were involved in mediating this phagocytosis. So first of all, we just looked at TAM receptor expression initially in those macrophages, and we showed that if you had TAM mutants, or by this time, we had developed floxed alleles for the TAM mutants, and we had a driver that would inactivate it specifically in microglia and not in other cells. And we could show that the TAM receptors, AXL and MER, but most specific, very importantly, MER, were actually important or critical for recognizing these dead cells in neurogenesis and for clearing them from the brain. So that was one of the ways we got back into the brain.
Yeah.
It's a longer story, but that's been the area where we've worked most, which is on the expression of the TAM receptors, MER and AXL, in these brain macrophages, microglia.
Yeah. So as we age, do the TAM receptors change their expression, or?
Well, that's a good question. As we age, I think, in general, there's a little bit of microgliosis, and so one of the TAM receptors is the TAM receptor that's constitutively expressed in all these phagocytic cells is MER. The other TAM receptor, AXL, is very sensitive to any inflammatory stimulus. So in most macrophages, its expression levels are actually quite low. In microglia, for example, in the brain, its expression level is very low. But if the microglia are responding to a traumatic injury or to a viral insult or to neurodegenerative disease, that's been the stuff we've worked on the last half dozen years a lot, then the AXL levels go way, way, way up. Oops. So they go way up in response to any inflammatory stimulus. So, and then this was found by people looking for markers of progression of neurodegenerative disease. So in Alzheimer's disease and Parkinson's disease, upregulated AXL expression in microglia, which you can actually detect in serum and in cerebrospinal fluid, turns out to be a very, very consistent and reliable marker of disease progression. So this AXL upregulation, it's quite dramatic. It's on the order of 20 to 40-fold increase in expression levels in the cells.
And it's in the CSF, you said?
It's in the CSF because it's AXL. All these proteins are cell surface receptors, but when the receptor gets activated, this is true for many receptor kinases, not just the TAMs. Downstream in the cell, it activates an atom-dependent protease that clips the ectodomain of the receptor off and liberates it, and that becomes a soluble protein. And then you can detect it in all kinds of body fluids. You can detect it in CSF, you can detect it in serum, in some settings where you can detect it in urine, you can detect it. So there are now actually good tests that can be performed on patients. So if a woman comes into the clinic and is in pregnancy and is experiencing preeclampsia, her AXL levels will go way up in serum. So
so
they can actually test this. They can test this in people,
Wow.
So in Alzheimer's disease, we were interested in testing that. And again, this is an example for how science can lead you in ways that you wouldn't expect. So a lot of the Alzheimer's community, Axel turned out to be, in the last 7 or 8 years, consistently turning up. People are paying more attention to Axel. We said, well, we've got all these mutants. We could cross the mutants into a mouse that, there are zillions of mouse Alzheimer's models now. Right? We can cross these mutants into a couple of these mice and see what happens. And so I had a student, Yutong Huang, very, very talented graduate student, who did this. And she discovered that, in fact, the TAM system is incredibly important for microglia to recognize the initial deposition of amyloid beta material in the brain. So
clears up the amyloid beta.
Well, that was the idea. I mean, and the way the system works, it gets down to a detail of the TAM system that's actually very important, that we also worked on in my lab over many years as a result of being prodded by Tony Hunter. So the TAM system, you got receptors and you got ligands that bind the receptors and activate the kinase activity in the receptor. That's the way RTKs typically work. But in the TAM system, you got one other component that is essential. So, and that is the presence of a phospholipid called phosphatidylserine, all right?
Yeah.
So the TAM ligands are actually bipartite. They've got a C-terminal part of the ligand, and we worked all of this out, that binds to the receptors very, very, very tightly, subnanomolar affinity. Then they have an amino-terminal part called a GLA domain, which binds to phosphatidylserine.
I see.
And so if you lop off this GLA domain of the ligand, it will still bind to the TAM receptors the same way as the full-length protein, but it's completely incapable of activating the kinase.
Oh.
So this, you have to have the GLA domain to bind to phosphatidylserine. So the TAM receptors and their ligands are together acting as a receptor for phosphatidylserine. Now, phosphatidylserine is a common phospholipid. It's in every single membrane of every cell in your body. But in all the normal cells, all the phosphatidylserine, the membrane is a bilayer, and all the phosphatidylserine is inside the cell facing the cytoplasm. So the external world never sees it.
Yeah.
So when cells get infected or they're going to die by apoptosis or by many other stimuli, you activate a set of scramblases which put phosphatidylserine on the surface of the cell. And now that's a signal for various things.
Eat me or something.
Eat me, or I'm infected, or I'm sick, or I'm not feeling well, or do something, But in general, it's called an eat me signal. It's the most common and the most powerful of the eat me signals by which dead cells are recognized by phagocytes. And that's how the TAM system works, because that's why it's so important. The TAM ligand, the aminoterminus of the TAM ligand binds to this externalized phosphatidylserine. The C-terminus of the TAM ligand binds to the TAM receptor on the phagocyte and engages the whole system.
Oh, okay.
So what Yutong showed was that in these forming wispy plaques, there's a lot of externalized phosphatidylserine.
Oh.
And that the microglia are actually using that all the plaques are decorated by Gas6 and protein S, the TAM ligands. That was not known beforehand.
Yeah.
And then the microglia use that to basically eat this stuff up. And so she was able, working with Axel Numion here at the Salk, to show if you don't have TAM receptors in microglia, their ability to do this phagocytosis is diminished tenfold. So it's a major drop, huge drop. And so we assumed that since it was thought that microglia were actually controlling the growth of these plaques, that we should have 10 times more plaques in the brain sooner, right?
Yeah.
When she actually went in and counted in the mice that didn't lack TAM receptors in the microglia, we didn't have more plaques, we had fewer. And so And so and then she did a whole bunch of experiments, published in a beautiful paper in 2021. What the system is actually doing. So the plaques, they're one of the defining features of Alzheimer's disease. We've known this for over a century. So Alzheimer himself developed these dyes that would bind to the plaques and stain them in tissue sections. But what you're actually staining are what are so-called dense core plaques, the highly compacted plaques.
Yeah.
The loose stuff does not stain by these dyes. And so what's actually happening is that what Yutong discovered is that the microglia are actually building the dense core plaques. So the dense core plaques do not form spontaneously. They're formed by microglia taking in the loose stuff into endosomes and then transferring those endosomes to lysosomes, compacting it there, and then either spitting the stuff out or dying, and that gets deposited in a dense core plaques. Because it's been known for a long time, for over 20 years, that there are two things that promote the formation of dense core plaques. One is micromolar concentrations of the A-beta peptide, and the other is low acidic pH.
Yeah,
right?
I heard that,
Right?
Low acidic pH.
Neither of those circumstances exist in the extracellular space of the brain, but both of them exist in lysosomes inside microglia. So lysosomes compact this stuff to very high density, so the protein's at very high concentrations now, and the pH drops. We've known the lysosomes that are acidic for like forever, right? And so this totally kind of, it reoriented people's thinking about the formation of these dense core plaques by 180 degrees. The microglia don't control their growth by phagocytosis, they make them by phagocytosis.
So that may explain why the plaques themselves don't necessarily correlate with dementia, because these are the dense core, but then the tiny ones that we're not seeing.
That's exactly right. Well, it's been known for a long time in vitro that the really toxic stuff of A-beta are these very small oligomers of the A-beta peptide. The A-beta peptide is it has varying lengths, but they're around 40 amino acids all. So if you make dimers, trimers, tetramers of those things, and you put those on neurons, and those are the really, really toxic stuff. So our idea now, which I have pushed just in the last year or so, is that these dense core plaques of Alzheimer's disease are really more equivalent to granulomas in other settings where your macrophages are confronted with something they can't get rid of. Like if you get something, you get stuck with, or like the classic example are microbacterial infections of lungs, like in tuberculosis. The tuberculosis bacteria is not moving all over your lungs. It's confined into these granulomas that are constructed by macrophages. And so we think I argue that the dense core plaques of Alzheimer's disease are exactly the same thing. So that's another situation where you just have to follow the experiments and where the data leads you. I can remember when Yutong was presenting the data initially, and she was showing me the quantification of the plaques, and I said, wait a minute, that's going in the opposite direction. What's going on? And you have to basically follow that up and, see what's what.
But you're having so much excitement. You said this paper was 2021, and then you have a new hypothesis. Then here's the big question. Then why did you decide to shut down the lab a couple of years ago?
Well, you put all this stuff out there, and the field will take it up, and so other people are going to follow up on it, and so I think that's part of the thing is fine. Oh, I see Janeway's immunology. That's exactly. That's the textbook I pulled down.
I had the old version. I just bought the new one.
Yeah, I had an old green. It wasn't red. It was green. I remember.
I had the green one.
Well, anytime. I mean, that project, that was her entire PhD thesis. So that was probably, by the time she left the lab and started her postdoc, it was 6 years. By the way, there's still a follow-up to that too. she also did the same kind of experiment in a mouse model of amyotrophic lateral sclerosis, ALS. And there, it's a really interesting phenotype. I mean, if you cross the MER mutants or the microglial-specific MER mutants into the ALS mice, you actually extend their lifespan by about a month or so. And this is a fairly rapidly developing model we're using. That's a long time.
Yeah.
And it turns out the reason for that is because when the motor neurons get sick in ALS, if you have a wild-type microglia, they will get sick. They're really metabolically stressed. They're getting lots of calcium influx. They will have phosphatidylserine all over their surface. And a wild-type microglia will kill the neuron by eating it alive.
Oh.
So the neuron is not yet dead. It still has connections to the muscle. It's still functioning not very well. It's really sick and unhealthy. The mouse is miserable, but the neuron's still alive. If you have a wild-type microglia, it will kill that neuron and eat it, and then you will lose the connection, and the mouse can't breathe and it will die. If you have a TAM mutant microglia, it can't eat the neuron, so the animal will actually, it's a miserable animal, but it will actually live longer. And we haven't published that. We're still going to publish it. So getting back to your question.
Yeah.
And I guess to get back to the first part of the thing, I know we can probably go. If I'm going to do some more experiments, then I have to gear up an operation. I have to plan on doing this for like another 5 or 6 years or so. So when I was young, when I was at MIT, I really had to decide at one point whether I was going to do science or whether I was going to do music. So I was a pretty good singer when I was young. That's actually it's a long story, but I did a lot of singing when I was young. And I decided, how much longer am I going to be around? I want to do something. Maybe I want to do something else. Maybe I want to get back into this. I got very involved with our local symphony orchestra here in San Diego. I helped to recruit our new music director, who was fantastic. I got more and more involved in that. A couple of years ago, they decided they were going to program the Verdi Requiem for their opening concert of the season. And this was a piece that I had sung a lot when I was a college student. Because when I was a college student, I sang with a big chorus in Boston called the Tanglewood Festival Chorus that sings with the Boston Symphony. And so I had sung with a lot of good musicians. And so I said, maybe I should sing in the chorus that's going to sing for this. So I had to audition.
You had to audition?
I had to audition. I had to audition. This is the first time I had auditioned in 41 years. So
in 40, I was joking that you were 41 years old.
No. I was in my late 60s at this point. I had to go. It's the first time I've auditioned for anything. So I did this audition, and the chorus that sings it here in San Diego is called the San Diego Master Chorale. So I had to sing with this. I auditioned for the San Diego Master. I sang with the bassist and the San Diego, and that was what really kind of convinced me that it was time to do this, because they let me sing. I passed with the audition, and I subsequently sang another concert with the orchestra.
Yeah.
On a Venezuelan piece at the end of last season. And if you're going to do that, and I'm going to do that, I've started writing music again.
Oh, you're writing also?
Yeah.
Music.
Yeah. And so, I decided it would be I would return. It was like a youthful thing. So that was kind of the basis of the decision. If I'm going to do this, I don't think I can. I have to do it now. So.
But you are also interested in architecture, wine.
Lots of things.
Many other things.
Lots of things. And I'm still maintaining some scientific connections. So there's a company that was started in Korea.
Excuse me.
Gesundheit.
Yeah.
That's sort of based on our work in the Alzheimer's thing. So what they've done is to take the TAM ligands.
Yeah.
I told you could lop off. We showed you could lop off the amino terminus, this glutamine. Yeah. The rest of that thing will still bind to the receptors with very, very high affinity, subnanomolar affinity, but it can't activate the kinase. And they replaced the glutamine now with a single chain antibody
Oh, okay.
to, the first test case, to A beta. So they've, so you make this single chain antibody, the variable heavy and light parts of the antibody, you make it as a single chain. You can link that on. And what they showed now is this now binds directly to A beta with the affinity that's determined by the antibody. And that hybrid protein activates the TAM receptor kinase and activates all the immunosuppressive functions of the TAM receptor. Because the other thing we showed that was really important is the TAMs are important for delivering immunosuppressive stimuli to dendritic cells and to macrophages. That's their other big job. And so this company is now up and running. They've raised a lot of money from investors in Korea. They published a beautiful paper in Nature Medicine showing that this proof of concept, this works fine. And in theory, they could put any antibody onto this, to any protein that they want to have microglia clear.
Yeah.
Or macrophages clear, right?
Yeah.
Anything. Well, it could be anything.
So that can be a lot of different applications.
Right. So they're initially going to try this for Alzheimer's, but they could do it for anything. And so I'm the head of the SEB for that company, and so I'm still maintaining. That's kind of fun too, because it's new science and it's potentially completely new therapies, and they're very, very clever people. So I'm still maintaining that.
And over the last several months, you have been the CSO.
I've been the CSO here, Salk.
Officer. And so you have come to full circle, like going from grad student, postdoc, faculty, chair of the academic council at Salk, and then CSO. And you have seen all the full spectrum. Any advice for the mid-career scientist or senior scientist like me? When should I stop? Or should senior scientists decide to stop like you did?
Well, most scientists don't do what I did. Most senior science, because this arguably almost the last paper we published is the most. Yeah. You can follow how tell you how things are cited now. It's for all the papers. It's probably it's one of the most rapidly cited papers.
Yeah,
in terms
so that's
of
why you
went into the literature. So most people don't do that.
You're living at your peak performance.
Yeah, so you got to decide what's best for you. I mean, for me, the thing really was I thought I might be a musician. I mean, it was like that was a tough thing. I mean, it was a tough decision to make. The guy who was my music professors at MIT and my writing professors, the composition professors, they were really, really good and they were encouraging, but. I don't know. We'll see how it works out. I thought I would be better as I could probably be more successful as a scientist than as a musician, but I don't know.
But another thing about you is you're extremely fit. there means you're late 60s, you said, and nobody would guess that you're late 60s. So what are the secrets?
You know this better than I do. I'm not going to give advice on this. Exercise. You got to exercise. Regular exercise. I did a lot of strength training at one point. I mean, it's come and gone. I mean, in terms of what I did. I think resistance training is actually, although, I'm not sure every doctor would necessarily agree this with old people, but it's still a reasonable thing for older people to do because you can decide how much you're going to do, right? You can adjust how much. It was always a good thing here because the gym at UCSD is 10-minute walk. You really don't have an excuse. Yeah. And not for getting regular exercise. For the longest time, I just got regular exercise. That was
for the new chapter in your life. We'll hopefully see you, and then once maybe you'll perform here.
Well, who knows? Maybe. Well,
this year,
some of my colleagues have. Tony Hunter went to hear me. I was only in the chorus, so you really can't necessarily assess my contribution. But Tony Hunter and Jenny Price went to hear me sing. I think maybe Jeff Wall was in the concert too. Yeah. These were concerts at the Rady Shell. With the new concert venue behind the convention center. Yeah.
Have you done any solo?
Not for big public things. No. Small recitals with my teacher. Yeah, but those are more okay.
Yeah. No, this is really. It was really nice to listen to all of this, how your scientific career took you through many different questions and how you kind of went back to the textbook in some cases. And,
Right.
These days, and this I think is a good inspiration for all of our young scientists, because sometimes they forget that you got to pursue where the biology takes you. It's not necessarily you should not stick to your expertise and just follow.
Right. I think you got to follow where the biology and the results of your experiments lead you. But more importantly, you've got to do things. You've got to basically do experiments that are allowing you to address an issue that you're fundamentally interested in solving. You got to have some question that you're really, really, you got to know the answer to. You've got to basically follow your curiosity. That's the most important thing. I mean, I see all these experiments now. A lot of the experiments that are done now involve technologies that are big team efforts.
Yeah.
So I see papers published, and I've been on some of those papers too because I participate in the team efforts, where I'm like, where you're like one of 40 authors on the paper. I mean, I would just not be interested in doing that. I mean, it's the particular science evolves, I guess, in certain ways. I can remember even when I was younger, experimental physics was kind of like that. You had to wait forever for your time on some big instrument in Europe, and then you ran your experiments and you spent 5 years analyzing them, and then when you publish the paper, there's a zoom analysis in the paper. So I wouldn't want to do that kind of science. I think you've got to really ask yourself fundamental things and follow them, and then maybe use new techniques to do that. I mean, I've always, for example, now in science, I think it's like a basic question. The signaling business with the phospholipids. Phosphatidylserine, as I said, it's about the most boring possible molecule. It's a lipid. It's a structural lipid that's literally in every single membrane. Yet your cells go through an incredible amount of effort to localize that lipid. Right?
Keep it inside.
Keep it in. Well, in that particular case, keep it inside. The enzymes so you need enzymes to do that because the energy barrier of doing this translocation through the hydrophobic core of the bilayer is too high. Those lipids can't translocate spontaneously. Yeah. The enzymes that do it are these giant 10 transmembrane proteins that look like calcium activated chloride channels. And in your genome, you have, again, I'm BSing you here. It's 18 or 19 genes that are of the scramblase family, right? That do the flip back out, that expose it. So you've got 19 of these enormous genes devoted. Then you have all the flip base, which set up the asymmetry initially. And there's a bunch of those too. There's something like 8 of those. Yeah. And so you can't just ignore that. That tells you that biology is using this signaling, this lipid signaling, to regulate all sorts of important things. And people have just basically ignored this for decades. That's a fascinating problem. I would really...
I think that this also comes back to your idea that the technology, and I think this is what I had heard maybe from Pete Souls, but I'm not sure. That technology expands your imagination. If you have the technology, then you can imagine that experiment. And now, the question that you're bringing up, one thing is, I don't know whether we have the technology to track this phospholipid.
That's a very good point. That's a very good point, because it's just developing. Because you think about what you're asking your technology to do. You can't just try to detect the phospholipid because it's in every cell. So you can't, for example, cut a section or allow access. You want something that allows you to detect the externalization, the presentation on the surface. And people are starting to develop it. There are proteins that bind to this. You can make them fluorescent. We've used some of those. but it's still very early days. It's very early. So there's going to be some technological development that's required for this. And, we know a lot of these genes now. We know the genes that are required to put those scramblers into the membranes. They're chaperones for that. So you can start mucking around with it genetically, but you're right. The technology there is still lagging behind what you need it to be to answer the question that you're interested in answering. You all right?
Thank you, Dr. Lemke.
You're welcome.
It was really wonderful, and I could have gone for another couple of hours just
I'm not sure what I told you.
talking
It's
about science.
relevant about performance, but
No, I think everything that we do in biomedical research is to increase our performance, whether it's physical, emotional, intellectual, and irrespective of age, sex, and health condition. So what we are talking about is, for example, the person who has this old, major Alzheimer's disease, for that person, peak performance may be just being able to remember the names of everybody in
their family, yeah.
That's performance for that particular person. Because we are always thinking performance is this muscle guys running on or lifting. That's not necessarily true. It's for every single stage. Performance means very different things.
Right.
So thank you so much.
My pleasure. Thank you.